Okay. So we're doing Punnett squares today, which I think a lot of people have some experience with. And like I said, they're quote unquote fun, I guess, fun for biology. This is about as fun as we get in biology, sadly. So I'm going to talk about what a Punnett square is, how to do it, and then I should share that with you guys on Zoom. And we'll do some practice Punnett squares as well. Yep, that's what, yeah, we're going to do. I have a worksheet to hand out today. So we'll practice that way. And I think I have another one that I can hand out as well as kind of extra practice. Sorry, my computer is being really slow. All right. Share, and here. Okay, so Punnett squares. Why do we use Punnett squares? Basically, it's just a way to figure out the ratios of phenotypes and genotypes of offspring. So you can work them if you know the parents' genotypes. We'll set it up that way at first, but you can also work backwards. So we'll do an example. If you know the offspring, then you can figure out what one of the parents' genotypes is. So you can work kind of forward or backwards. We're going to do both ways. Essentially kind of what Mendel was doing way back in the day, but in square form. So the easiest way to learn these is just to do some examples. So I'm going to jump in with an example that I have laid out on the screen, and then we will go to a worksheet that I have for you guys to work on for a little bit, and you'll do the same thing essentially. And we'll talk about that after you guys get done with it. Okay, so we're going to start with one character. We're going with the P theme, P color. Yellow is going to be dominant. Green is going to be recessive. So we have a big Y and a little y. And we're going to cross what we call true breeding individuals. I don't think I've defined this yet. I can't remember. But they are essentially homozygous. So true breeding just means they're homozygous. They don't have, there's not one that's heterozygous, obviously. So we have a big Y, big Y. That's going to be one parent. And then we're going to cross that with an individual that has two little y alleles. So this is the parental generation is big Y by little y, little y. Okay. Now, so the F1 generation is what we're looking at, right? We're crossing these true breeding individuals, these parents, with each other. So here we have on the top of the, so when you draw out a Punnett square, for this first part, it's just going to be, we're looking at one character, right? Just P color. We'll get into looking at two characters at one time. That's in the next PowerPoint that I'll bring up. But for now, we're just going to look at the one character, P color. So you'll set up essentially a square divided into 4 individual quarters. So you have one parent on the top. So parents go along the top or along the side. So big Y, this is going to be this parent. And the alleles are going to be separated, right? Because either one can be passed down to the offspring. And then our other parent, our green parent, little y, is going to be on the side here. So you write out all of the alleles on the top and the side of the Punnett square. And then you just look within each square at what alleles are donated. So you can imagine each of these quarters within this bigger square is one offspring. So these are all the possibilities of the genotypes and phenotypes of the offspring between these two parents. So if we look at this first upper left square, you have a big Y from this parent and a little y from this parent. So those come together to make a big Y, little y individual. So for you guys on Zoom, this big Y and this little y combine to give us a big Y, little y. So that's a heterozygote, right? There's a dominant and a recessive allele. because we're dealing with just simple dominance, a simple dominance scenario, it's going to be a yellow P. So that individual will have yellow P's. Because you have a big Y, it totally masks the little y, the green. And then you just do the same for the other three. So here we have a big Y being donated in the upper right by the parent on top, a little y being donated, see a big Y, little y. You can probably see the pattern that's going to occur when you have two true breeding individuals where each of them can only donate one type of allele. If you have a dominant and a recessive parent, you're going to have all heterozygotes as a result. They're all going to be big Y, little y, because they have to get one allele from, say, their father, one allele from the mother. So genotype, all heterozygous, big Y, little y. And make sure with each of these Punnett squares, you can tell me what the genotypes are, as well as the genotype ratio. So here it would be 4 to 0. So 4 heterozygotes to 0 of anything else. The phenotype ratio is going to be the same because they're all yellow. Remember, phenotype is what the offspring look like. So 4 yellow to 0 green. That's a really straightforward Punnett square. So I want to do that one first so you guys can kind of see how you can reason out what the offspring are going to be like. Any questions about that? All right. So now, oh, whoops. Forgot to have the other names on here. Yeah, so the result is always the same. I'm going to go over a few ratios that you want to basically remember. This is one of them. So you're always going to have all heterozygotes when you have two true breeding individuals. When you have one dominant, one recessive, it's always going to be the same pattern. And you can see why based on the Punnett square. And then there's a couple others that I'll point out and mention. You definitely want to remember these ratios. So 4 to 0 is going to be your true breeding individuals where you have one dominant, one recessive parent. Okay. So then if we cross, let's say we go one generation further, we're going to cross 2 of those heterozygotes. With plants, this is fine. They're related. Humans, not so much. So we're going to cross 2 F1 individuals. So that looks like this. You have a heterozygote and a heterozygote, because that's all the offspring we had from the previous one, right? So those are that's the only cross option you have if you want to use 2 F1 individuals. So now when you cross 2 heterozygotes, you have a little bit of variability because now each of them has a big Y and a little y to donate. So in the upper left here, so we put big Y, little y on the top, big Y, little y on the side for the parents. Here we get a homozygous dominant, yellow, in the upper left, because those are 2 big Y alleles that are being donated here. Over here, now we have a little y in the upper right, along with a big Y on the left side here. So that's going to be a heterozygote. Big Y, little y. Same for down here. So it's hopefully it's pretty obvious how we're doing this, just pulling each of the alleles into those squares. So big Y, little y, we get another heterozygote. And then finally, we get a little y and a little y in the bottom right quadrant. So that's our homozygous recessive individual. We finally have a green P, because now each of these parents can donate a little y. And if they do, it results in a green P. So the genotypes here I've listed. You have 3 different genotype options. So before we just had the one. We had big Y, little y. Now we have, oh, sorry. Yeah, the letters. So the genotypes are the specific alleles, the letters. So we have one big Y, two of the heterozygotes, big Y, little y, and then one homozygous recessive. So then the phenotypes, now you just look at what the expression is. You're not really worried about, well, you should worry about the letters in that you need to figure out what they look like. So if you didn't have this yellow color here, you want to figure, you'd know that anything with a big Y is going to be yellow. So you have 3 that are going to turn out yellow because they have at least one big Y. And then you have 1 green, so 3 to 1. 3 yellow to 1 green is our phenotype ratio. I think I have that written out. Yeah. So this is what that looks like. A genotype ratio, and this is always going to be the same when you have 2 heterozygotes crossing. This is a ratio combo that you want to remember. Genotypes 1 to 2 to 1. That means 1 homozygous dominant, 2 heterozygotes, 1 homozygous recessive. 1 to 2 to 1. But phenotypes, we only have 2 different colors, right? We have yellow and we have green. So you have a 3 to 1 phenotype ratio, 3 yellows to 1 green. This is called, I don't think I have this up. Okay, yeah. Again, always the same ratios. So just as a reminder, these are always going to be the same when you have 2 heterozygotes mating. This is called a monohybrid cross. So mono means 1, so we're talking about 1 character, just P color. Hybrid means you have 2 heterozygotes crossing. So this is a monohybrid cross. When you have a monohybrid cross, you have this genotype and phenotype ratio every single time, assuming we're dealing with a simple system like this. We're going to talk about dihybrid crosses in just a minute, where we are actually looking at 2 different characters at one time. So those Punnett squares get a little bit bigger. So if you understand this, you'll be able to figure out the dihybrid cross as well. But let's practice with this first to make sure you guys kind of understand this concept. So for you guys on Zoom, there's a sheet called Punnett Problems 1. It should look, at least the first page looks something like that. So go ahead and open that up and you can start working on it. I'm going to let you guys work on it at first. You can work on it in class with other people, and then we'll go over the answers together. Make sure you guys understand it. So there's a front and a back. So I already laid out the Punnett squares for you. You just have to put in the parents and the offspring and figure out those ratios. Okay, so for number 1, we have the blue eyes, brown eyes issue. So when I set up my Punnett square, what do I put across the top? What are one of the parents? For number 1. Yeah, exactly. And both the parents are the same, right? Yeah. So big B, little b. And really, it doesn't matter what order you put any of these in, as long as you have 1 parent and 1 parent. You can't mix up the parents. In this case, it wouldn't really matter. So you could put the little b's first if you wanted to. So what goes in this Punnett square in the upper left? Yeah, big B, right? So you have these 2 coming together. And then here we have what? Yeah, big and little. Here we have same, right? Big and little.
2.
Yeah, 2 littles. Okay, so that's gonna be our Punnett square for, and we just did that, right? What is that called? Do you guys remember what I called it? Mono something?
Mono, we didn't have hybrid.
Yeah,
Yeah.
mono hybrid cross. So one characteristic we're looking at, but each of them are hybrids, so heterozygotes. Okay. So then what does that, so great, you can make the Punnett square. The important part is figuring out the ratio essentially of these offspring. So how many homozygous dominants are there? One, right? So we have one homozygous dominant. How many heterozygotes? Yep, so one here and one here. So 1 to 2. And then how many recessive? 1. Yeah.
So that's how you want us to write it out.
Yep. So the ratios are written out like this. And when you're dealing with one character, you'll either have 3 numbers if you have a heterozygote, or like I said, if you have just, when we had all of the same, basically it was 4 to 0. So it might only be 2 numbers. But in this case, this is going to be the homozygous dominant. The second number is going to be the heterozygous. And then this number is going to be homozygous recessive. So when you see 3 numbers like that, that's what the 3 numbers mean. So 1 to 2 to 1. And then the phenotype ratio, how many brown-eyed individuals do we have? 3, right? So we have big B, big B, big B. So phenotype would be 3 to 1. Yeah. So 3 to 1. We have 1 recessive. So this is going to be the dominant. And we don't care if it's homozygous or not, right? We're just looking at what the offspring look like. And then this 1 would be the recessive. So 3 to 1. So when you see that 1 to 2 to 1 and that 3 to 1, you should know, okay, that's a monohybrid cross. So if I told you the offspring has this ratio of 1 to 2 to 1, the genotype ratio, you should be able to figure out what the parents are based on that, because they're both heterozygotes, right? Same with the 3 to 1.
And that will always be the case?
Yes, that'll, there are some exceptions that I'll let you know. I have to let you know if it's a simple dominance situation. So in a simple dominance, yes. There's a couple of more complicated things that we'll get into. But yes, I'll let you know what the system is. And then if it's just simple, straightforward dominance, that's the case. Yeah.
Is that only looking at the 3 ratio? Or like if we also look at the 2, like just the 3 to 1, could you also infer?
Yes. Yep. Both of them. So 3 to 1 and 1 2 to 1. Good question. So the genotype and the phenotype ratios for a monohybrid cross, or they're always going to represent a monohybrid cross if we have just a straightforward system. Good questions. Okay, so that's number 1, right? Yeah, we got all those. And then the second one, a little bit different. The only difference really is that one of the parents, or the parents are a little bit different. I'll leave this set up. Okay, so what do we got? We have red flower and white flower. So now what are our parents? What is the genotype of the red flower? Well, I gave it to you, didn't I? So yeah, big R, little r. So we have one parent that's heterozygous, and on an exam, I might not tell you that it's big R, little r. I might just say you have one heterozygote as a parent, and you should be able to, whatever letters you want to use, you should be able to figure that out. And then you have one homozygous recessive, right? So little r, little r. All right, so I think everyone understands how to put these squares together, right? So I'm just going to go through and do it. So we have a big R and a little r here. We have 2 little r's. We have a big R, little r. Then we have 2 littles. Okay. So now what is our, so yeah, how many homozygous dominants are there? 0, right? We don't have any homozygous dominant. How many heterozygotes? 2. Big R, little r. We have 2 heterozygotes. And how many homozygous recessive? 2, right? So I guess you would put, yeah, so for the ratio, you could do 0 to 2 to 2. That's not a ratio you need to remember. I won't ask you to work backwards from there. The 1 to 2 to 1 ratio, I will, but be able to tell me what the ratio is, but you don't have to memorize that one, as long as you can figure it out with Punnett squares. And then the phenotype ratio, how many red individuals do we have? 2, right here. And then how many white individuals? 2, yeah. So that's going to be a 2 to 2 ratio. And here, red, white. Okay, so that's just slightly different. It's not always 2 heterozygotes crossing. All right, any questions about that? Okay, now the fun one's next. We're going to work backwards. So working backwards, I gave you a few bullet points to kind of help you think about how to work backwards here. So we have, so albinism is a recessive condition. So you have to have 2 recessive genes in order to be an albino, to have albino skin color. So let's see, we have a woman who is an albino. The mom is albino, married to a non-albino man, and they have an albino child. So we want to figure out the genotype of the father. So let's see, we'll put the mom on the side here. So what's her genotype if she's albino?
2 little.
Yeah, 2 little A's, yeah. Okay, and then the father,
He's mixed. Something.
he could be big A though, right?
No.
Good job. Okay, so we'd start out, if you're not sure what the heck we're doing, good job though. We know he has at least 1 big A, right? So we don't know what that one is, but you guys have figured it out because you're super smart. So we know we have to have 1 albino child. So that's going to be little a. So here we know we know he has a big A because he's not albino, right? But he could have a big A here and a little a. So big A, little a, big A, little a. So neither of these kids are going to be albino. They're going to have non-albino skin color. Over here, if he had a big A, we'll just do the opposite. That would be the same. We'd have big A, little a, big A, little a. So not possible, right? Because we don't have any little a's in here. So then he must be heterozygous. So we must have a little a. So we have little a, little a, little a, little a. So they essentially have a 50% chance of having an albino offspring, right? So half of their kids would be carriers for it, and half would actually have albino skin color. Okay, awesome. So if you understand that concept of working backwards, which you guys were really quick at, that was awesome. That's about the amount of detail that or the most difficult type of working backwards I would ask you to do in that situation. Any questions about that?
For step 2, was it the recessive A that you did not see?
Yes, yeah, exactly. You must have the recessive A. Okay. So yeah, Punnett squares, I think they're pretty approachable and understandable for people, which I really like, especially after the biochemistry we've been doing. So hopefully you guys agree with that. Whoops. So I'm going to go into now some dihybrid stuff. So we're going to look at 2 characters at once. And then I won't have time to get into it today, but we'll talk about some kind of weird scenarios where you don't really have this totally complete, obvious dominance, which is where it actually kind of gets more interesting. All right. So I'm going to pull up the next PowerPoint. All right, so I'm going to talk, well, wait for this to come up. I'm going to talk at first a little bit more about Mendel's laws. So if you remember, Mendel had two laws that we looked at, the law of segregation and the law of independent assortment. And we're going to look at how that plays into the single character crosses and the two character crosses, which we haven't gotten into yet. Okay, so first off, okay, we know what this is. We're crossing two pea plants with different alleles, right? So we have the mother and the father. First off, what are we doing when we put two alleles together here? Do you remember what that process is called? What creates a zygote from two gametes? Going back to meiosis, basically. That starts with an F. When a sperm and an egg come together, what is that called? Fertilization. Yeah. So what we're representing here in each of these squares is just fertilization. So the father plant fertilizing the mother plant, for example. And then this Punnett square. So this simple one character Punnett square. Do you guys remember which of those two laws this essentially represents? I know it's a tough question, remembering those two laws. So segregation or independent assortment. We're dealing with alleles, not whole genes.
Independent.
Other. We're going to talk about it. Thank you for answering. So this is the law of segregation. So this is the law of segregation because when we're talking about the law of segregation, we're talking about alleles from one gene. So here we're just looking at flower color. That's the only gene we're looking at. You have two different alleles, big P and little p. Those two alleles from each parent will segregate from each other during meiosis. So that's the law of segregation. When you see law of segregation, think segregation of alleles. So alleles. Yeah, exactly. So this P doesn't necessarily have to go with this P, right? So this isn't the only option. This P can go with this P as well. So they can mix and match in basically any possible way. It's all just, it comes down to chance. So law of segregation is a segregation of alleles. They're not linked to each other. It's not like a big P has to go with a big P or vice versa from the two parents. Okay, so law of segregation. And then here I have the definitions of the monohybrid. Well, not definitions, but a reminder of the monohybrid cross. We're only dealing with one character. So A in this case, one character, two alleles, but one character, say P color. When we get into the dihybrid cross, we're dealing with two characters in one square. So we're going to go from 4 possible individuals to 16. So we're going to make the Punnett square a lot bigger, but it's the same concept as long as you know how to write out the parents on each side and you understand how the alleles come together. So I'll show you that in a minute. But two characters, dihybrid. And we're still dealing, so dihybrid, specifically the hybrid part refers to two heterozygotes. Yeah, exactly. Each of the gametes, which is essentially one of those squares, the offspring squares that represent the gamete, will have one allele, I'm sorry, those are the offspring. The gametes are going to contribute one allele from each character or gene. So all of the offspring are going to have 2 alleles for each of those characters. So if we look at, these are going to be the parents that we're going to cross. So this one can donate essentially one allele from each of the genes. So what are we talking about? Color and then texture of the P. So yellow or green, and then smooth versus wrinkled. So there's a Y and an R option. This parent is going to donate one of each, and all he has is a big Y and a big R. So that's all he can donate. And then the mom, we'll say this is the mom. All she can donate is a little Y and a little R. but they each only donate one of each, right? They're not, because we have the meiosis is basically cutting the amount of DNA in half. So that when these get fertilized, now the offspring have the right amount of DNA. So we'll look at what this essentially looks like on the next slide in a dihybrid situation, a dihybrid Punnett square. Oh, whoops. I messed up my animations. Okay, so we have these gametes coming together. So we're gonna have a dihybrid, sorry, heterozygous for both options. Okay, so that's what we're gonna cross. We're essentially going to cross a big Y, little y, big R, little r, by big Y, little y, big R, little r. So we're gonna do a dihybrid cross. So imagine we're crossing two of these. I should have written this up there, but big Y, big R, little r, by the same thing. We're crossing two of these. Okay, so you can see how it gets a bit more complicated. Don't worry about these quarters. That kind of just adds confusion. But if we look at the individual options for the gametes of the father, so he can donate a big Y and a big R. So that's this option. He can donate a big Y and a little r. He can donate a little y and a big R. So basically every possibility. And then he can donate a little y and a little r. But notice there's only one y and one r in each gamete. That's where it can get a little bit confusing. You can't have two y's or two r's in a gamete. And then the mom can do the same thing. Does that make sense so far? Okay. So then if you work that out, which I'm not going to take the time, it's already up here. I'm not going to take the time to actually work it out, but I'll show you how. I'll pick out a couple random squares and show you that it's just the same as with the monohybrid cross. You just have to take into account two different characters, a Y and an R, instead of just a Y, for example. So if we start, we'll start here in the upper left. So the dad is donating a big Y. The mom is donating a big Y. So you have two big Ys. And then they're each also donating a big R. So you have big R, big R. If we pick kind of a mixed one, we'll go here. So for you guys on Zoom, looking at this one, the little y, big R, big R. So we've got the mom donating a little y, the dad donating a little y. Boom, two little Ys. And then each of them are donating a big R, big R, big R. So that's going to mean a green but smooth P. So you have yellow and smooth, you have yellow and wrinkled. You have green and smooth, green and wrinkled. So the green wrinkled's like the super duper recessive, all little alleles, Ys and Rs. And this is showing the other law of Mendel's. So we talked about the law of segregation being the fact that alleles are separating from each other. This is showing that genes are also separating from each other randomly. So that's called the law of independent assortment. In other words, just because a P is yellow doesn't mean it has to be smooth. It can be yellow and wrinkled as well. So the two characters aren't linked to each other. That's what the law of independent assortment shows. And that's why you have to have two characters in order to kind of show that. Just because a P is wrinkled doesn't mean it has to be green. Or just because a P is green, it doesn't have to be wrinkled.
So when there's multiple like outcomes, like two different types of outcomes, so it's independent.
Independent assortment, yeah. So when you have two characters, two genes, it's independent assortment. Yep. If you're talking about one gene, you're talking about the law of segregation. or principle of segregation. I don't remember what he calls it, law or principle. All right, so we're not going to worry about the genotypic ratio for a dihybrid cross because it's really long. There's a lot of different possible genotypes, like the combinations of letters. The ratio you want to remember for a dihybrid cross is this 9:3 to 3:1. That basically means that you have 9 individuals. If you count these all up in the Punnett square, you'll see this. You have 9 that are homozygous dominant. You have 3 heterozygotes, but each kind of heterozygote. So it could be heterozygous for either character. So you have homozygous dominant to the heterozygotes, and then you only have one homozygous recessive. Little y, little y, little r, little r. And that's the phenotype. It's not the genotype. So 9:3 to 3:1. If you see that phenotype ratio, you know you have a dihybrid cross. You have 2 individuals that are heterozygous for 2 different characters that are crossing. Oh, wow. And that is all the time we have. So I'll get into some of the more complicated or stuff that doesn't fit those patterns on Wednesday.